Welcome back to the deep dive. Today, we are doing something a little different.
Yeah.
Usually, we took a pretty broad look at a topic, epidemiology, public health, the big shifting plates of medicine.
The 10,000-foot view.
Exactly. But today, we're going microscopic. We are rolling up our sleeves and walking into what I kind of think of as the engine room of modern medicine.
The engine room. I like that image.
Right.
It implies noise, heat, essential machinery, and probably a bit of grease.
It feels appropriate. We spend so much time in medical education and honestly on this show talking about the clinician at the bedside.
Mm-hmm. The detective?
The detective looking for clues, listening to the heart, feeling the abdomen, taking the history. But I've been reading through Murray's Medical Microbiology, specifically chapter 16, and it really struck me.
What's that?
If the clinician is the detective, the microbiology lab is the forensics team.
That is the perfect analogy.
You think so?
Oh, absolutely. Because you can have the best detective in the world, Sherlock Holmes standing right there at the bedside. But if they mishandle the evidence before it even gets to the lab,
the case goes cold.
the case goes cold. Or worse, you convict the wrong suspect.
And that is exactly what we are unpacking today. We're breaking down the laboratory diagnosis of bacterial diseases.
It's a huge topic.
It is. We're using Murray chapter 16 as our spine, but I've also pulled some technical supplements from Jowett's chapter 47 just to flesh out some of the detail.
Good call. Jowett's is great for the nitty-gritty.
And honestly, the big theme that's jumping out at me isn't about the shiny new technology, though we will get there.
Right.
It's a simple, almost brutal truth. Garbage in, garbage out.
It is the golden rule of the laboratory. It's the one thing that keeps microbiologists up at night.
Really?
It doesn't matter if you have $1 million Malikoff mass spectrometer or the most advanced high-throughput PCR machine on the planet.
Yeah.
If the specimen you put into it was collected poorly or transported slowly or, and this is a big one, was just a swat of spit when you needed deep lung sputum,
then the result is worthless.
it's worse than worthless.
See, that's interesting because I think a lot of us assume the black box model.
Explain what you mean by that.
You put the sample in the tube, send it away in the pneumatic tube system, and the truth with a capital T just comes back on the computer screen a few hours later.
And that's a very dangerous assumption.
Yeah.
A bad result is actually worse than no result.
How so?
Well, if I tell you no growth, you might at least keep looking for another cause. But if I give you a false positive because of a contaminant, you might treat a bug that isn't even there. You blast the patient with vancomycin, destroy their kidneys, and you completely miss the actual infection that's killing them.
And the flip side.
The flip side is just as bad. You send a swab that dried out in transit, I tell you no growth, and you confidently stop antibiotics on a patient who actually has a raging anaerobic infection.
So our mission today is to dismantle that black box. We are going to build a mental map of the whole process.
I like that. A mental map.
We're going to walk through the body, system by system blood, brain, lungs, urine, stool, and look at how you get from patient with a fever to specific organism identified.
From suspicion to certainty.
And for everyone listening who is prepping for exams, single best answer, or MCQs, you need to pay attention. Chapter 16 is an absolute goldmine for questions because it deals with process errors.
And process errors are, without a doubt, the most common reason for diagnostic failure.
It's not the bugs tricking us.
No, we trick ourselves.
Okay, let's start with, I think, the highest stakes environment, the blood.
Right. Sepsis. This is where the clock is ticking loudest.
The scenario is classic. You've got a patient, they're spiking fevers, maybe hypotensive, heart is racing, white count is through the roof. You suspect bacteria in the blood. Exactly. And Murray makes a really interesting point here about the numbers game. I think the lay intuition, and maybe even the student intuition, is that if someone is septic, literally dying of a blood infection, their blood must be teeming with bacteria.
Like a swarm of bees, you'd think.
Right. But that's not the case.
Not at all. It's a very common misconception. In reality, in a septic adult, the density of bacteria is incredibly low.
How low are we talking?
We're often talking less than one organism per milliliter of blood. Sometimes as low as one organism per 10 milliliters.
Less than 1 per ml. That just seems impossibly low for something that's causing systemic collapse.
It is low, but remember, the symptoms of sepsis are largely driven by the host's massive immune response to the bacteria, not just the sheer volume of bugs. It's the cytokine storm.
Okay, that makes sense. But for the lab, this creates a massive statistical problem.
A huge problem. If you just draw a standard 5 milliliter syringe of blood, Statistically, you might not even scoop up a single bacterium.
You're trying to catch a single fish in a massive lake with a coffee cup.
That's a great way to put it. You'll probably come up empty.
So if you miss the fish, the lab reports no growth, and the clinician on the floor thinks, oh, okay, it's not sepsis.
Exactly. And they stop the antibiotics or they go chasing some red herring. This is why volume is the single most important variable in blood cultures.
The single most important.
Without a doubt. The data from Murray is stark on this. They state that collecting 20 milliliters of blood yields 40% more positive results than collecting just 10 milliliters.
40%. That is a massive difference in diagnostic yield. That's almost a coin flip difference in finding the cause.
It's huge. It could be the difference between life and death. So the standard protocol for an adult is roughly 20 milliliters per culture set.
Per set. And what does a set mean?
A set is usually 2 bottles. That 20 milliliters of blood gets divided. You put about 10 milliliters into an aerobic bottle.
For oxygen-loving bugs like Pseudomonas.
Right. And you put the other 10 milliliter into an anaerobic bottle for those bugs that hate oxygen, like bacteroids from the gut.
Okay, so if I'm the intern on the ward drawing the blood, I need to be greedy. I need to fill those bottles.
You do. Don't underfill them.
But what about kids? You can't drain 20 mils of blood out of a neonate without causing iatrogenic anemia.
No, absolutely not. Pediatrics is always the exception. For children, it's all weight-based. It might be anywhere from 1 to 5 milliliters total.
And that's okay for them.
It is, partly because their bacterial loads in sepsis can actually be much higher than in adults, which helps offset the lower volume. But for adults, volume is king.
Okay, so it's got the volume, but let's talk about the other great enemy of blood cultures, the skin.
Ah, yes. The skin. It is a bacterial zoo.
Even on the cleanest person.
It doesn't matter. It is covered in Staphylococcus epidermidis, Cutibacterium, Micrococcus, a whole ecosystem. The needle has to pass through that zoo to get to the sterile vein.
And if you drag some of those skin bacteria along for the ride and into the bottle,
the lab grows it.
Yeah.
And suddenly the doctor gets a call at 3 a.m. saying the patient is growing staph. They think it's sepsis when it was really just, a dirty needle stick.
That's a false positive, a contaminant.
Exactly. And this isn't a trivial problem. It's incredibly expensive. One study showed that a single contaminated blood culture adds, on average, about 3 extra days to a hospital stay.
3 days?
And thousands of dollars in costs because we treat just in case. We give them powerful antibiotics they don't need.
All because of a bad wipe.
All because of a bad wipe. And Jawitz gives us the technical detail on how to prevent this. It's not just a quick swipe with an alcohol pad like you're giving a flu shot.
No.
It's a 2-step prep. You want 70% alcohol first to clean off the surface oils and dirt, to scrub. Then you follow that with an antiseptic like iodine or, more commonly now, chlorhexidine. That's what actually disinfects the skin.
And there's a timing element here too, right? I feel like this is where people get impatient and cut corners.
Yes. The infamous dry time. You have to let the antiseptic dry completely. If the chlorhexidine is still wet when you stick the needle in, it hasn't finished killing the bacteria. It needs that contact time.
Makes sense. Now, even with perfect prep, contamination still happens sometimes.
It does.
So how does the clinician or the student taking an exam distinguish a true positive from a contaminant? If the lab calls and says, we have staph epidermidis, how do I know if I should panic or just, ignore it?
It comes back to the numbers game and simple probability. This is why we never draw just one set of blood cultures.
Ah, you draw multiple sets.
You have to. You draw 2 or ideally 3 separate sets of blood cultures from different sites.
Mm-hmm.
So one from the left arm, one from the right arm, maybe a third one an hour later.
Okay.
Now let's say Staphylococcus epidermidis, a common skin bug,
Yeah.
grows in only 1 bottle out of 4 or 6 total bottles.
Just one.
The probability is that it was just a contaminant. It was on the skin at that one puncture site and nowhere else.
But if it grows in the left arm bottle, AHD the right arm bottle.
Now that's a pattern. That strongly suggests it's not just on the skin in 2 different places. It's actually circulating in the bloodstream. That's a true infection.
Even if it's a wimpy bug like staph epi.
Even then, if it's in multiple bottles drawn from multiple sites, you have to take it seriously. It could be a line infection or endocarditis.
So multiple sets are absolutely crucial for interpretation.
Essential. A single blood culture set is rarely enough to go on. You typically want 2 to 3 sets per septic event.
Got it. One last thing on blood before we move up to the brain. Timing.
Okay.
I've heard people say you have to catch the fever spike. Like, wait until the patient is shivering and shaking, then pounce and draw the blood.
Right. The rigors.
Yeah. Is that true?
Murray actually busts this myth pretty definitively. The fever spike corresponds to the release of cytokines, the IL-1 and TNF-alpha.
The immune response.
Which happens after the bacteria have entered the blood and been recognized by the immune system. The peak bacteremia might have actually been an hour ago.
So you missed the boat.
You might have. Practically speaking, you shouldn't wait. If you suspect sepsis, you draw the culture. Period.
Yeah.
And ideally, you do it before you start the first dose of antibiotics.
That's the critical part, isn't it? Before the drug.
It is the most critical part of timing.
Yeah.
Because once those antibiotics are on board, they start inhibiting bacterial growth almost immediately.
Even if they don't kill the bug instantly.
Right. They could just stun it. It won't grow in our culture bottle. Your chance of a positive culture just drops off a cliff after that first dose.
Okay, so to recap blood, volume is king, prep the skin properly, draw multiple sets from different sites, and always draw before the drugs.
Perfect summary.
Let's move to the central nervous system. Cerebrospinal fluid, or CSF.
Now, this is a true medical emergency. Bacterial meningitis kills fast.
How fast?
We are talking hours. A patient can go from I have a stiff neck and a headache to permanent brain damage or death in less than a day.
So the lab has to be fast.
The whole system has to be fast. And the sources are very, very specific about how to handle this fluid once you tap it from the spine. There is a do-not-do rule here that seems counterintuitive to anyone who cooks or, buys groceries. You mean the refrigeration rule?
Yes. We are taught that biological samples rot, so we should put them in the fridge to preserve them. But for CSF...
Never, ever refrigerate CSF if you are looking for bacteria.
Why? What actually happens in the fridge to the sample?
The two biggest killers in bacterial meningitis, Neisseria meningitis and Streptococcus pneumonia, are incredibly fragile, fastidious organisms.
Fussy.
Very fussy. And Neisseria in particular is what we call autolytic.
Autolytic. Self-breaking.
Essentially, yes. It contains enzymes that break down its own cell wall. When it gets cold or runs out of nutrients or just gets stressed, these enzymes kick in. The bacteria effectively commit suicide.
That is incredibly dramatic. It's like a spy swallowing a cyanide pill if captured.
It is. The cell just disintegrates. So if you put that tube of CSF in the fridge overnight, you come back in the morning to a sterile culture. The evidence has literally destroyed itself.
And in the meantime, the patient is dying of untreated meningitis, and you're scratching your head saying, but the culture was negative.
Exactly. It's a catastrophic error.
So what is the protocol then?
It goes straight to the bench. Do not pass go. Do not collect $200. You walk it to the lab. You hand it to a human being.
And once it's there...
Once it's there, because the bacterial numbers can be low, just like in blood, we use a centrifuge.
The spin.
We spin it down hard to concentrate any bacteria, any white blood cells, into a little pellet at the bottom of the tube. Then we take that pellet and smear it on a slide for the Gram stain.
Which gives us that immediate visual clue. And for the exam takers, we absolutely need to know what we are looking at.
You do.
So if we see Gram-negative diplococci...
That's Neisseria meningitis. They look like little pink kidney bean-shaped pears.
Mm-hmm.
And often you see them inside the white blood cells, the neutrophils, because the cells are trying to eat them.
And if we see Gram-positive diplococci...
That's Streptococcus pneumonia. They'll be purple. And they're described as lancet-shaped pears, like little spear tips pointing at each other.
That's a great visual. No, culture is the gold standard, but Murray mentions that we are seeing a shift here, too, similar to other areas.
Yes, the shift towards Nafion nucleic acid amplification tests.
PCR.
Exactly. The BioFire panels are a great example. They are so much faster. You can get an answer in about an hour. And they can detect the bugs even if the patient has already had a dose of antibiotics that killed the live bacteria.
How does that work? Is it because the DNA is still floating around?
That's it, exactly. The antibiotic might kill the organism so it can't grow, but its DNA is still there. It's like a ghost of the bacteria.
PCR can find the ghost.
PCR can find the ghost. Culture needs a living, breathing being.
That is a fantastic way to put it. Okay, let's slide over to other sterile fluids. We're talking pleural fluid in the chest, synovial fluid in the joints, peritoneal fluid in the belly.
The principles here are very similar to blood and CSF. These are spaces that should be completely sterile. If anything grows, it's almost always significant.
And what is the deep rule here? I feel like we are about to introduce a villain in our story.
The villain is the cotton swab.
Oh.
The rule is this: volume is your friend, and swabs are your enemy.
Why is the expert community so anti-swab? It just seems so convenient. Just dab it.
It is convenient for the clinician, but it is an absolute nightmare for the lab.
Yeah.
Imagine you have a patient with a joint infection, a septic knee. It's full of pus.
Okay.
If you just dip a little cotton swab in it and send it to the lab, you're setting yourself up for failure for I'd say 3 main reasons.
Okay, hit me. What are they?
Reason 1, volume. A swab holds maybe zero 0.1 milliliters of fluid if you're lucky. We just talked about how bacterial numbers can be low. You need more volume to find them.
Okay, volume. What's 2?
Reason 2, entrapment. Cotton fibers are like a net. They trap the bacteria. You can rub that swab all over the agar plate, but half the bacteria stay stuck in the cotton. They love the cotton.
They don't elute off.
Exactly. And 3.
What's the third one?
3 is the big one, especially for deep infections. Oxygen. Swabs expose the sample to air.
And anaerobes hate air.
It's toxic to them. If you have an anaerobic infection, a bug like bacteroides or fusobacterium, that swab is a death sentence for the bacteria. You've killed the suspects before they even reach the station.
So what is the alternative? What should be done?
Aspirate. Always aspirate. Use a needle and syringe. Drop the fluid as much as you can possibly and safely get, and then either cap the syringe to keep the air out or inject it into a sterile, air-free tube.
And that gives the lab so much more to work with.
So much more. If you send me 10 milliliters of fluid, I can spin it, I can do a gram stain, I can culture it aerobically, anaerobically, I can set up fungal stains, I can save some for PCR. If you send me a swab, I can barely do a gram stain.
That is a very clear exam tip. If the question asks for the best specimen for an abscess or a fluid collection, and one of the options is swab and the other is aspiration.
Always choose aspiration. Reject the swab.
Okay, moving on. Let's talk about the respiratory tract. This seems trickier because, unlike the blood or the spine, the entryway, the mouth, is absolutely filthy with bacteria.
It is. The mouth is a bacterial metropolis. Billions of organisms. Hundreds of species. Streptococci, actinomyces, anaerobes, you name it.
So we have to divide this into the upper and lower respiratory tracts.
Exactly. You have to be very precise.
Let's start at the top. Upper respiratory, pharyngitis, a simple sore throat.
Right. And most of the time it's viral, but the one we're really looking for is group A, Streptococcus. Streptococcus pyogenes.
Because of the risk of rheumatic fever?
Precisely.
So the technique here seems simple, swab the throat. But there is a nuance, isn't there? Specificity matters.
It matters a great deal. You want to vigorously swab the tonsils and the posterior pharynx, any areas that look red or have pus on them.
And what do you not want to touch?
You do not want to touch the tongue, the cheeks, or the teeth.
Why is that? Is it just to avoid making the patient gag?
Well, nobody likes gagging.
Yeah.
But biologically, the tongue and cheeks are covered in different bacteria and, more importantly, saliva.
And saliva is a problem.
Saliva contains enzymes, like lysozyme, that can actually inhibit the growth of strep. And it contains all this confounding flora that will overgrow the plate and make it hard to find the real pathogen.
So you want the sample from the site of inflammation, not the commute to get there.
Perfect way to put it. Now, Murray also mentions a special case. If you see a pseudomembrane, maybe you're suspecting diphtheria.
Which is rare now, but still exists.
You should actually lift the edge of that membrane and swab underneath it to get the best yield.
Good tip. Now, there is a very specific and very serious safety warning in the text regarding the upper respiratory tract. It's about epiglottitis.
Oh, this is critical. This is a do not touch situation.
Yeah.
A true medical commandment.
Explain the scenario.
If you have a child, it's usually a child, though adults can get it too, who is drooling, leaning forward in that tripod position, struggling to breathe, voice is muffled.
And you suspect the epiglottis, that little flap in the throat, is swollen and infected.
Do you swab it to confirm the diagnosis?
I'm guessing the answer is no.
The answer is absolutely not. Never.
Why?
Because the epiglottis is acting like a hair trigger trap. The airway is almost completely blocked. The trauma of the swab or even a tongue depressor can trigger a complete laryngospasm. The muscle seize up.
And the airway snaps shut.
Completely. You can convert a sick child into a dead child in seconds, right there in the exam room.
That is terrifying. So how do you diagnose it if you can't touch it?
You back off. You get them to a controlled setting like an operating room to secure the airway if needed. And you rely on blood cultures.
Because the bug is in the blood, too.
The bug, which is usually Haemophilus influenza type B in unimmunized kids, is almost always in the blood as well. So you diagnose it from the blood culture, do not touch the throat.
A very high-yield safety tip there. What about sinuses? We treat sinusitis all the time with antibiotics. Z-Pax, amoxicillin?
We do, but we almost never diagnose it microbiologically.
Because we can't get to the sinuses easily.
Exactly. A nasopharyngeal swab, sticking a swab way back in the nose, tells you nothing about what is actually growing inside the sinus cavities. The bacteria are totally different.
So what's the right way to sample it?
To do it properly, You would need to stick a needle through the cartilage of the face directly into the sinus cavity and aspirate the fluid.
Which sounds both invasive and incredibly painful.
It is. So it's very rarely done. We just treat empirically. We guess based on the likely suspects: pneumococcus, Haemophilus, Moraxella.
Let's go deeper then. The lower respiratory tract, pneumonia. This brings us to the spit versus sputum debate.
This is the bane of the microbiology lab's existence. I cannot tell you how many cups of spit I have had to reject in my career.
Explain the problem for our listeners. Why is it so hard?
The problem is that to get a sample from the lungs, it usually has to pass through the mouth on the way out.
And the mouth, as we said, is dirty.
So the patient coughs, they spit in a cup, and they send it to us. The lab's first job is to figure out, is this pus from the deep lung, or is it just saliva from their mouth?
And how do you do that? Do you just, look at it? Can you tell by eye?
Not reliably.
Yeah.
We have to look under the microscope. We do a gram stain of the raw sample, and we use a scoring system, often based on the Bartlett criteria. We are looking for cells.
What kind of cells?
Specifically, we're looking for squamous epithelial cells.
The skin cells of the mouth.
Exactly. Squamous cells are those big, flat, pavement-like cells that line your cheek and tongue.
Mm-hmm.
They are not found deep in the alveoli of the lungs.
So if you see a lot of them.
If we look at the sample under the microscope and see a ton of squamous cells, we know this is saliva. It didn't come from the lungs.
So garbage in.
Garbage out. We reject the sample. We call the floor and say, this is an unacceptable specimen. Please collect another. We don't even culture it.
Why not? Why not? Just see what grows.
Because if we did, we'd just grow a mix of normal oral flora, and the doctor might see that report and start treating the wrong bug, while the real pathogen in a lung goes untreated.
So what does a good sample look like under the scope?
A good sample has lots of polymorphonuclear leukocytes, PMNs, white blood cells. Because an infection, a pneumonia in the lung, summons a flood of white blood cells to fight it.
So lots of WBCs, very few epithelial cells.
That's the magic combination. That tells us this is active inflammation from the deep airway. That's a sample worth processing.
Now, what if the patient can't cough up a good sample? Or what if you are specifically worried about anaerobic pneumonia?
Anaerobes are very tricky because they live normally in the mouth and the gums. So any expectorated sample is useless for diagnosing anaerobic pneumonia. It's always contaminated.
So you can't use a sputum sample for that?
Never. If you really need to diagnose anaerobic pneumonia, you have to bypass the mouth entirely.
How do you do that? Teleportation.
Close. Bronchoscopy. You go down with the scope. But even the scope has to pass through the mouth and pharynx. So they use a special tool called a protected specimen brush.
What is that?
It's like a tiny brush inside a catheter, which is inside another catheter. It's nested. You keep it covered until you are deep in the target lung segment. Then you pop it out, grab the sample, pull it back into its sheath, and withdraw the whole thing.
Wow. It's like a little James Bond gadget for getting a clean sample.
It is. It's the only way to reliably diagnose anaerobic pneumonia.
Incredible. Okay, let's move to the head and neck, ears and eyes.
The rules are similar here. It's all about surface versus deep.
Let's start with otitis media, middle ear infection. Kids get this constantly.
And just like sinusitis, we almost never culture it.
Why not?
Because the infection is behind the eardrum. To get a sample, you'd have to perform a tympanocentesis.
Sticking a needle through the eardrum.
Which parents and kids typically frown upon. It's painful and invasive.
So we just treat empirically based on the likely pathogens.
Right. But if it's otitis externa summer's ear, that's in the external canal. That's on the outside. A swab is perfectly acceptable there. You're looking for Pseudomonas or Staph aureus.
Makes sense. What about the eye?
The eye is very high stakes because it's so delicate and vision is on the line. If you have a deep infection inside the eyeball, endophthalmitis.
That sounds terrible.
It is. That requires a needle aspirate of the vitreous or aqueous fluid. But for surface infections like conjunctivitis or a corneal ulcer, swabs or gentle scrapings work.
And Murray mentions a specific timing issue here too, right, for certain bugs.
Yes. Some of the pathogens that cause eye infections, like Neisseria gonorrhea in a newborn's conjunctivitis, are very fastidious. They die fast once they leave the body.
So you can't just put the swab in a transport tube.
The recommendation is often to inoculate the culture plate at the bedside.
You take the agar plate to the patient's room.
Exactly. You take the swab and you immediately streak it onto the plate. We actually use chocolate agar for this. It's not actual chocolate, it's just cooked blood agar, which is very nutrient-rich. It gives the bug its best chance to survive.
Moving down the body to arguably the most common sample the lab receives, urine.
Oh, by far. It is the highest volume sample, and it is fraught with collection and transport issues.
The basic anatomy logic is straightforward. The bladder itself is sterile, but the distal urethra, the exit pipe, is colonized with bacteria from the skin.
Right. So every time you urinate, you're potentially flushing some of those normal urethral bacteria into the collection cup.
Hence the invention of the clean catch midstream technique.
Right. The theory is you clean the area first to reduce the surface bacteria. Then you start voiding into the toilet to flush out the urethra. That's the first portion. Then, without stopping the stream, you catch the middle portion in the cup.
And that theoretically represents the pure bladder urine.
That's the theory. In practice, asking a sick, elderly, or confused patient to perform this rather acrobatic feat is optimistic.
It's messy.
It can be very messy, but let's assume we get a decent sample. The next hurdle is transport. Transport is a huge deal for urine.
Huge, because bacteria absolutely love urine. It is essentially a warm nutrient broth for them.
That's a perfect growth medium. And Murray gives a terrifying statistic about E. coli. It can double its population every 20 minutes at room temperature.
Okay, so let's do that math. Let's say you have a patient with a tiny, clinically insignificant number of bacteria from contamination, say 1,000 organisms per ml.
Okay.
And a nurse leaves that cup on the counter for 2 hours before it gets to the lab.
Well, 2 hours is 6 20-minute doubling times. So that 1,000 becomes 2,000, then 4,000, 8,000, 16,000, 32,000, 64,000.
Suddenly, it looks like a raging infection.
It looks like a significant UTI when it was nothing.
Garbage in, garbage out.
The quintessential example. So the rule is refrigerate immediately. Cold stops the doubling. Or use a special collection tube that contains a preservative, like boric acid, that essentially freezes the bacterial population in time.
And when we finally get the result, we use the 10 to the 5 rule for interpretation, right?
Generally, yes. The lab does a quantitative culture. We count the colony-forming units, or CFUs. If there are more than 100,000 or 105 CFUs per milliliter, that's traditionally considered indicative of an active infection, like cystitis or pyelonephritis.
But there's a big caveat for symptomatic patients.
Always treat the patient, not the number. This is critical. If a young woman has classic symptoms, burning, urgency, dysuria, but the colony count is only 123 or 103 DARs, that can still be very significant.
So the 10 to the 5 rule isn't absolute?
Not at all. It was originally developed for screening for asymptomatic bacteriuria in pregnant women. For a symptomatic patient, any number of uropathogens can be the real deal.
Okay, that's a key clinical point. Let's talk about genital specimens. This field has changed more rapidly than almost any other.
Oh, absolutely. This is the story of the shift from culture to molecular diagnostics.
And we were focusing on the big two here, Neisseria gonorrhea and chlamydia trachomatis.
The main causes of STIs like urethritis and cervicitis.
In the old days, say 20 years ago, we had to culture them.
And it was a nightmare. Gonorrhea is incredibly fussy. It dies if you look at it wrong. It needs special media, special transport conditions, and chlamydia is even worse.
Why is that?
Because chlamydia is an obligate intracellular parasite. It can't live on its own. It needs to grow inside of actual living human cells like a virus. You can't just streak it on a plate. It required complex and expensive cell culture techniques.
So what do we do now?
NATEs. Nucleic acid amplification tests,
Yeah.
PCR. They are now the undisputed standard of care. They are vastly more sensitive and specific than culture ever was.
And the specimen collection is much easier, I understand.
Much easier. For men, you don't even need an invasive urethral swab anymore. You can use urine.
But wait, hold on. For a UTI, you just told us we have to throw away the first part of the urine and collect the midstream.
Exactly. And this is a super common point of confusion and error.
So explain the difference.
For a UTI, the infection is in the bladder. So we want the bladder urine. We flush the urethra first to get rid of contaminants. For gonorrhea and chlamydia, the infection is in the urethra.
Ah, so you want the urethral cells.
You would wash those infected cells and organisms out. So for an STI test, you collect the first void urine, the very first 15-20 milliliter of the stream.
That is a crucial high-yield distinction. UTI equals midstream. STI equals first void.
Spot on. Get that wrong, and you could miss the diagnosis.
What about syphilis? Treponema pallidum.
This is the great unculturable pathogen. You cannot grow it on a standard laboratory plate. It just won't grow.
Still, with all our technology.
Still. So how do we diagnose it?
Yeah.
If you have a primary lesion, a chancre, You can scrape the base of it and use a special technique called dark field microscopy.
Which sounds like something from a sci-fi movie.
It looks like one, too. The background of the microscope is black, and the live bacteria light up like these beautiful, eerie silver corkscrews spinning and flexing around.
But most labs don't have that anymore.
No, it's a dying art. So for almost all cases, we rely on serology. We test the patient's blood for antibodies their bodies has made against the syphilis bacteria.
Okay, we're in the home stretch of the body systems. Fecal specimens, stool.
Ah, the ultimate diagnostic challenge. This is the needle in a haystack problem.
Because stool is mostly bacteria to begin with.
Stool is basically 100% bacteria by dry weight. It's overwhelmingly normal, healthy flora. We are looking for the one bad actor, the one pathogen, hiding in a crowd of 100 billion bystanders.
Transport must matter here, too.
It does, but for a different reason than urine. Some key pathogens, like Shigella, are very sensitive to the acid produced by all the other bacteria in the stool.
So the normal flora can kill off the pathogen in the tube.
Exactly. So we use Carey-Blair transport media. It's essentially a buffer. It doesn't help anything grow. It just keeps the pH neutral so the sensitive bugs like Shigella can survive the trip to the lab.
And similar to some other tests, you have to tell the lab what you're looking for. You can't just send a sample for stool culture.
Right. A standard routine stool culture is designed to look for the big three: Salmonella, Shigella, and Campylobacter. But if you suspect, say, Vibrio cholerae in a patient with rice water stool,
you need to ask for it specifically.
You have to tell us. We need to use a special agar called TCBS thiosulfate citrate bile salt sucrose. It turns yellow if Vibrio is there. If you don't ask for it, the lab will never set it up and you will miss the diagnosis.
And then there's the big one in hospitals, C. diff, Clostridioides difficile.
Yes. And the rule here is counterintuitive but absolute. Do not culture.
Do not culture. That sounds so wrong for a microbiology deep dive.
I know, but finding the organism itself doesn't prove disease. A large percentage of hospitalized patients are asymptomatically colonized with C. diff. It's just living in their gut, not causing any problems.
So if you culture it, you'll find the bug, but you don't know if it's the cause of the diarrhea.
Exactly. You need to prove the toxin is present. The toxin is the weapon that causes the damage and the colitis. So we test for the toxin itself, usually with an EIA, or we test for the toxin gene using PCR. We're looking for the weapon, not just the suspect.
That makes perfect sense. Okay, before we wrap up with susceptibility testing, let's briefly touch on identification. We've grown the bug on our plate. Now, how do we figure out what it is?
It all starts with the gram stain. Is it purple, which is gram-positive, or pink, which is gram-negative?
Right.
Is it a dot, a coccus, or a rod, a bacillus? How are they arranged?
Murray has a fantastic table on this table 16.3. Let's do a quick rapid-fire association game for the listeners.
Go for it.
Gram-positive cocci in clusters.
That's Staphylococcus. The clusters look like bunches of grapes. If it's then coagulus positive, it's S. aureus, the really bad one.
Gram-positive cocci in chains.
That's Streptococcus. They look like a string of pearls.
Gram-negative rod, and it ferments lactose on a McConkey plate, turning it pink.
That's a classic enteric, one of your gut bugs like E. coli or Klebsiella.
Okay, a gram-negative rod that does not ferment lactose and has a sweet sort of grape-like or corn tortilla-like smell.
That is the one and only Pseudomonas aeruginosa. The grape smell is actually a distinct chemical it releases, 2-aminoacetophenone. You can smell it through the plate.
Now, that's the old school way. Biochemicals, smelling plates. But the lab is changing fast.
Enter MEL-DFAF,
which stands for
Matrix-Assisted Laser Desorption Ionization, Time-of-Flight Mass Spectrometry.
It sounds like a weapon from Star Wars.
It kind of acts like one. You take a single colony of bacteria, smear it on a metal target, and you blast it with a laser.
You blast it with a laser?
Yes. It vaporizes the bacterial proteins into a charged cloud, and then comes the time-of-flight part. An electric field accelerates those proteins down a long vacuum tube. A detector at the end measures how fast they fly.
And what does that tell you?
The small proteins fly fast. The big proteins fly slow. It creates a unique spectral fingerprint based on the protein profile of that organism. The computer then matches that fingerprint to a massive database of thousands of bugs.
Can you get a species-level identification in?
Minutes, instead of the days it used to take with biochemical tests. It has revolutionized the workflow. You know exactly what you are fighting almost immediately after the colony grows on the plate.
It's incredible. Finally, let's get to section 11. Antimicrobial susceptibility. We know what the bug is. Now, how do we kill it?
This is the guide to therapy. This is how we move from empiric guessing to targeted definite therapy.
The outline mentions two main methods: broth dilution and disc diffusion.
Right. Broth microdilution is the gold standard. You have little wells with the bacteria and increasing amounts of an antibiotic. The lowest concentration that stops the bug from growing is the MIC, the minimum inhibitory concentration.
So if the MIC is very low, the drug is very potent against that bug.
Correct. A low MIC is what you want to see.
And disc diffusion is the Kirby-Bauer method, the one we see pictures of all the time.
Yeah, you have the agar plate covered in bacteria, and you put down little paper discs infused with different antibiotics.
And you measure the zone of inhibition.
The clear circle around the disc where the bacteria can't grow. A bigger circle means the bug is more susceptible to that drug.
But, and here's the big clinical warning, pharmacokinetics matter. An S for susceptible in the Petri dish doesn't always mean it's going to lead to a cure in the patient.
This is a fundamentally important concept. We mentioned it briefly before, but the drug have to actually get to the site of infection in a high enough concentration.
Can you give an example?
Aminoglycosides, like gentamicin, are great drugs against many gram-negatives, but they will not work in an anaerobic abscess.
Why not?
Because they need oxygen to be actively pumped into the bacterial cell. It's an active transport mechanism that requires O2. In an abscess with no oxygen, the drug can't get in. The bug is susceptible on the plate, but functionally resistant inside the abscess.
So context is absolutely everything.
Always. You have to think about whether the drug can cross the blood-brain barrier for meningitis, or if it gets concentrated in the urine for a UTI.
So to summarize the whole journey, the lab is not a black box. It's a crucial partner in care. But it needs good data to work with.
Garbage in, garbage out. Volume matters. Transport matters. Aseptic technique matters. And knowing what to specifically ask for matters.
Before we hit the recall quiz, here's a final provocative thought to chew on. We are moving more and more toward culture-independent tests. PCR, NECs, they are incredibly fast and sensitive. They tell us who is there from their DNA.
Right.
But they don't give us a live bug.
And if you don't have a live bug, you can't test which antibiotics will kill it.
Exactly. So is that the trade-off?
That is the double-edged sword. If we lose the culture, we lose the phenotypic susceptibility profile. We might know it's MRSA because we found the MEK gene, but we won't know if it's also resistant to clindamycin or trimsulfa unless we actually grow it and test it.
So are we trading precision in identification for a new kind of blindness in treatment planning? Something for everyone to think about as these technologies evolve.
Indeed. It's a huge question for the future of microbiology.
All right, pause your walk, put down the coffee. It's time for the recall quiz. 5 questions, single best answer style. I'll ask the question, give you a moment, then our expert will give the answer and the why.
Let's do it.
Question 1. You are managing a patient with suspected sepsis. You draw 5 millimillis of blood into a single aerobic bottle. The lab reports no growth after 5 days. Why is the lab likely to have missed the diagnosis? Take a second to think about that.
The answer is inadequate volume. For an adult, you need approximately 20 millimels per culture set. Bacteremia is often a very low-density state, frequently less than 1 organism per ml of blood. A 5 millimole sample is simply not enough to statistically guarantee that you'll even catch one single bacterium.
Okay, question 2. A sputum sample arrives at the lab. The technician performs a gram stain on it and reports many squamous epithelial cells. The lab then rejects the sample. Why? What's the reason for that rejection?
The answer is saliva contamination. Squamous epithelial cells are the cells that line the mouth and cheeks, not the deep airways of the lungs. A good sputum sample should be full of neutrophils or PMNs and have very few epithelial cells. A sample with many squamous cells is just spit, not sputum.
Right. Question 3. You have a patient with severe watery diarrhea after a long course of antibiotics. You suspect C. difficile colitis. You send a stool sample for a stool culture. The lab calls and says, this is the wrong test. Why? Why is culture wrong for C. diff?
The answer is that culture detects the organism, but not its ability to produce toxin. Many non-toxigenic strains of C. diff exist and can harmlessly colonize the gut. To diagnose the disease, you must specifically detect the toxin itself, using a test like an EIA or the toxin gene, using a NAD or PCR.
Excellent. Question 4. A 4-year-old child presents to the emergency room with drooling, a high fever, stridor, and is sitting in the tripod posture. You suspect epiglottitis. Why must you not attempt to swab the throat? What's the critical safety reason here?
The answer is the risk of inducing complete airway collapse. In epiglottitis, the airway is already critically narrowed by the swollen epiglottis. The trauma of a swab or even a tongue depressor can trigger a laryngospasm, fully closing the airway. Diagnosis should be made in a controlled setting, and you should rely on blood cultures for the microbiology.
And the final question, question 5. You see a painless, indurated ulcer, a classic chancre, on a patient's genitals, and you suspect primary syphilis. You swab it and ask the lab for a routine culture. Will it grow? Can you culture syphilis?
The answer is no. Treponema pallidum, the spirochete that causes syphilis, cannot be grown on cell-free artificial media. It is unculturable by standard methods. The diagnosis is made either by dark field microscopy of the lesion exudate or, much more commonly, by serology.
And that is a wrap on Murray Chapter 16, a fantastic tour of the engine room.
Good luck with the studies. And remember, ask for it, don't swab.
We'll see you on the next deep dive.